So now it's time for our final presenter for this session. Dr. Jan Frederik Stevens received his master's in pharmacy and pharmacy license and PhD in medicinal chemistry from Groningen University in the Netherlands. He received postdoctoral training at Oregon State University and the Free University of Amsterdam and Leibniz. Oh, now I'm learning German or Dutch. The Leibniz Institute for Plant Biochemistry, Halle, Saal, Germany. And as he introduced himself, he is now an associate professor of medicinal chemistry in the College of Pharmacy at Oregon State University. He is also a principal investigator in the Linus Pauling Institute at the same university. Mass spectrometry-based metabolomics is a new direction in his lab for discovery of biological effects of vitamins and phytochemicals. With this, I present Dr. Stevens. Yeah.
I am going to talk about a single natural product which is called Xanthohumol. And Xanthohumol contains two names. Xanthos is from Greek, it means yellow, and the humol is from Humulus, and that is the genus name where this compound is from. It's Humulus lupulus, that is commonly known as the hops, from the hops that is an ingredient of beer. Now we started to work on this back in the mid-1990s, and I published a paper in Phytochemistry in 1997 publishing a few compounds, flavonoids from the hops. And then later we published more, and now with other people, there are now maybe 25 known flavonoids from the hops. The main principal compound in hops is the Xanthohumol. Now, the brewers, they have been using hops for millennia, I would say, but the brewers never paid any attention to this compound. It was first described in the Journal of Chemistry in 1913, so it's now 100 years old. It was identified in the 60s, and still the brewers didn't pay any attention to this because it had no taste. Now we, in the 90s, figured out that it had cancer chemopreventive activities, and we also identified the compound in beer, and that is shown here on this slide with, you will now see there's a kind of a primitive LC-MS equipment at that time, and we were the first to identify these compounds in beer. Now that got the interest of the brewers, and then we got funded to look at the biological properties of Xanthohumol. Now here's the structure of Xanthohumol. You see this big compound there. It's a chalcone. It's a flavonoid compound. It has a C5 tail. It's the prenyl group. And the prenyl group has been shown to be very important for the biological activity. Now we're not interested anymore in cancer chemopreventive activities because it is kind of boring research, because you need a long time, and if it works well, you don't see anything, right? So we shifted our focus more on things that you could measure in the short term, and we became interested in metabolic syndrome. Now, what is metabolic syndrome? Metabolic syndrome is not really a disease. An MD will not tell you that this is a disease, but it is a cluster of risk factors. And you can have genetic factors, you can have overnutrition, that is just a simple word meaning you eat too much, lack of physical activity, cigarette smoking, and what you can get out of these input, you can get insulin resistance, elevated blood pressure, dyslipidemia, central obesity, and chronic inflammation. Now all of this, if you have two of these, or three of these, depending on the definition, you're diagnosed with metabolic syndrome, you have an enhanced risk for developing type 2 diabetes and cardiovascular disease. Now I also want to mention that, of course, in the United States, about a third of the population is obese, and about a quarter of the US adult population has been diagnosed with metabolic syndrome. So this is a huge, huge problem, and we cannot treat all those people with FDA-approved drugs. Now we did a study in rats. This is the Zucker rat, and that is a model for type 2 diabetes and metabolic syndrome. So we had 4 groups of 12 rats each, and we gave the first 12 rats a 0 dose. The second group, we gave them 20 milligrams. Now this is not the 20 milligrams that we gave the rats, this is the scale dose to humans. So if we would give a human a 20 milligram dose, we would scale it back to the rats, calling allometric interspecies scaling according to the FDA formula. Now we gave then a 3 times higher dose, 60 milligrams, and then 180 milligrams. Now these rats, they were first put for 3 weeks on a high fat diet, and then for the last 3 weeks on a normal fat diet. And when we submitted this proposal to the NIH, we only looked at body weight, fasting plasma glucose, cholesterol, insulin, triglycerides. And I am very glad that I included the metabolome profile, because that is where we actually identified the mechanism of action. Now, talking about metabolomics, the name was coined in, I believe it was in the mid-1990s. But actually, the pioneer of metabolomics was at Oregon State University, Linus Pauling, who received a Nobel Prize for chemistry and also for the Peace Prize in 1964, but he did much, much more. So he published in 1974, so 20 years before metabolomics became known as metabolomics, he published a paper called Orthomolecular Diagnosis. If you read this paper, this is nothing else that we would call today metabolomics. Now today, of course, we have much, much better equipment to do this. In our lab, we have 12 mass spectrometers for all kinds, and one mass spectrometer I like in particular. This is the so-called ToF. This is a QToF instrument, and it can do very fast scanning, 100 spectra per second, and it can do combined with UPLC. Now what you get, if you have many, many samples that you analyze, in a run of about 40 minutes, you can see here, we get about 8,000 peaks. 8,000 peaks, that is quite a number of peaks. So actually what we're doing is, in the old days we were testing a single hypothesis with a single endpoint. Now all of a sudden we're testing 80,000 hypotheses. And after that we can pick and choose and see which one is the best one. Now of course when you do this, you've got to be careful, because if you have these 8,000 peaks, you've got to make sure that the first sample and the 100th sample, that the peaks that you call in, let's say, the first run, you call it, let's say phenylalanine, then in the 100th run, you've got to still call it phenylalanine, otherwise your whole method is worth nothing. So this is called the data processing and peak. Here on the right side, you can see data pre-processing, so you get many of the overlapping chromatograms, and we pick the peaks, we align them, so there's an algorithm for that, so that we call each peak the same one. So here are the peaks are aligned, so this is a single peak that we pick out of that. And of course, if you do this for all of these 8,000 peaks, you get a big, big data matrix, and can see that these are the samples and the metabolites. And then, of course, you can do a lot of statistics and data visualization. Now, on the left side, you can see you can do principal components analysis. This is a way to reduce all that information into a two-dimensional plot and see if the samples that you're comparing are different from each other. And then finally, if you identify peaks that are of interest, then of course you want to know what they are, because then the metabolites need to fit in a metabolic pathway, and then you can draw conclusions from your analysis. Now here, when you do this, of course, you've got to be careful that if you try to relate dose with effect, you also got to make sure that the compound that you're testing is bioavailable. Now that is shown here. The control, of course, there was no Xanthohumol in the test system, so there we measure no Xanthohumol. Now in the middle, in the low and the middle dose groups, you can see that there is an increase in the plasma steady state levels, and in the highest dose you can see that the plasma concentration reaches about a micromolar, almost a micromolar, 0.9 micromolar at the highest dose. So this is good, and the two middle groups, of course there was a difference, but difference was not significantly different. Now then we look at the classical endpoints, and this is body weight gain. So this is a model for obesity and weight loss. So these rats, the Zucker rat, it grows continuously throughout life. So it never reaches a steady state. So they grow all the time. Now you can see that at the top curve, this one here, this is the control. So they have the highest body weight at the end of the study, and the highest dose it has the lowest body weight, and that decrease or the attenuation of the body weight is about 14%. So that's quite significant. And what is also significant is that the fasting plasma glucose drops over the dose levels by about 25%. So what we here is have an anti-obesity compound with an anti-glycemic effect. But now, of course, is what is the mechanism of this activity? That's the big question. You could test one hypothesis after the other, and you could spend probably 3 PhD students on it, and they get very frustrated if they can't show what is the true hypothesis. So we do a different way. So we do metabolomics analysis. So we measure these 8,000 peaks that I've shown you. Now, out of that, we do a statistical test, and we look which ones are significantly different in abundance between the groups. Now, out of there, we distilled 133 metabolites using the t-test, and we did that the 0 versus the highest dose level, and then we determined a Pearson's correlations test. Now, what is that? Now, I got to give a little example here, because if you have, let's say, metabolite X here in the, this here, metabolite X and metabolite Y. Now, if they across the dose levels both increase, that means that Xanthohumol is inducing a particular pathway that increases both metabolites X and Y, and that could, for instance, indicate that they're both on the same metabolic pathway. Yeah? Now, on the other hand, is an example, if across the dose levels there is an inverse correlation between metabolite X and Y, that could mean that, let's say, metabolite X is converted into metabolite Y. And from that, you can also draw conclusions, and I give that a red color. And if in this case you do a very simple data matrix, you get something like this, where green is there's a positive correlation, and in the red color there's an inverse correlation, and where there's black, there is no difference. Now, this is the result of that Pearson's correlation matrix. And you can see that, of course, we have grouped all those. So here are the 133 metabolites on this axis, and the very same metabolites are plotted on the x-axis, and you can see how all these metabolites correlate with each other. Now, this is very good because it will tell us right away, visually, where the action is. Where is something changing? Now, you can see here, for instance, what is sticking out like a sore thumb is acyl carnitine. See this big green area? That is really significant. You can see phosphatidylethylamines. You can see the dicarboxy fatty acids. And here, this is a really highlighted area. There's a hydroperoxy fatty acids, and of course the fatty acids. So these are the metabolites we got to be looking at to look at the effect, the mechanistic effect of Xanthohumol. Now, let's first look at the dicarboxy fatty acids. And of course, I always tell the students, well, what do I need to know if you want to join your group and do metabolomics? I say, you've got to know your biochemistry, because without biochemistry, you can't do this work. But of course, I lie a little bit because I'm a pharmacist, so I got to learn biochemistry myself. Now, what's going on with fatty acids? If there is too much fatty acid, the body tries to get rid of that, and it does that by omega hydroxylation and then omega oxidation, and then it puts a carboxyl group at the tail end, so you get a dicarboxy fatty acid, and that is excreted in the urine. So this is a way for the body to get rid of overload of fatty acids. You can see that with treatment, so here is the 0 line, and this is the change, so Xanthohumol versus the control. You can see that if we treat the animals, there is, let's say, about a 40% reduction in the levels of these dicarboxy fatty acids. That is quite significant. Now, the same is true for the hydroperoxy fatty acids, and of course we know hydroperoxy fatty acids, they're products of reactions of reactive oxygen species with fatty acids, and they are bad because they can cause DNA and protein damage. And also you can see here that there is about 40% reduction in these hydroperoxy fatty acids with Xanthohumol treatment. Now, this is the most significant result in when I first saw that, I couldn't believe what I saw, but it is really true. Now, let's first start with the fatty acid. We all know that fatty acids, and these don't like to see fatty acids in the blood. because that means that it is causing insulin insensitivity, so insulin resistance. But here are acylcarnitines. See that? For the whole range that we can measure by mass spectrometry, there is now anywhere, let's say, from 5% all the way to minus 60% reduction in the levels of medium chain acylcarnitines. Now, what does that mean? Thinking about this for a long time, we need to think about metabolic overload, lipid overload. What happens if you eat too much fat? And that is what it says here. And of course, you have diet, you get fatty acids. This is the fatty acid overload. And at some point, you get these fatty acids are converted into long-chain acyl-CoAs. And of course, these are is the fuel for beta oxidation. So this is where your energy is coming from. Now you can see if there's pressure because there's overload, this beta oxidation pathway there is pressure, right? Because downstream, the acyl-CoA feeding into the tricarboxy, the TCA cycle, the Krebs cycle, of course this feeds into the electron transfer chain and eventually get ATP production. Well, if this part of the metabolism is saturated, so there is no more capacity, and you start pushing long-chain CoAs into this pipeline, of course, at some point, this is a leaking garden hose. It starts coming out as medium-chain acyl-CoAs, and of course, these medium-chain acyl-CoAs get converted back into medium-chain acylcarnitines. These are products of incomplete beta oxidation, and that, as you will see in the plasma. Now that is what I'm saying here. The fatty acids are converted to gamma, the omega dihydroxy fatty acids, and the long-chain CoAs convert into medium-chain acylcarnitines. Now the effect of seeing that with Xanthohumol through our metabolomic screen, that all of these products are greatly reduced when we treat our rats with this compound. So the conclusion from this is you can hypothesize that Xanthohumol improves beta oxidation of fatty acids. So you get reduction of acylcarnitine levels. That's good because this is also a biomarker of type 2 diabetes. You get increased oxygen consumption because that is shown here. You get more production of ATP. Of course, that takes more oxygen to be consumed. And you get increased energy expenditure. Now that explains why the rats that we treated weighed less. And you eventually get an anti-obesity effect. Now, what is the effect? What is the hypothesis? What is the mechanism for this? Now, we hypothesize that Xanthohumol uncouples the respiration from ATP synthesis by carrying protons across the inner mitochondrial membrane. And this is a fancy word for this is the protonophoric effect. So the electron transport chain is trying to be very efficient. It tries to use the proton gradient to produce ATP. And all we're doing now is we're carrying protons across the membrane by losing the proton gradient, and the mitochondria, they try to keep the membrane potential to a certain level, so they start being more active. So the mitochondrial activity increases, and that's what we want, because if you increase mitochondrial activity, then you also get more beta oxidation and more fuel is burned up. Now, of course, this is all fine, but of course, we should not forget this is all from the metabolomics analysis. So you've got to have another method for testing this hypothesis. And for that, we have, of course, the Seahorse extracellular flux analyzer. This is just a very fancy instrument, which is much liked by PhD students because you get very fast data. So what you do is you grow your cells on a 96 well plate, and you titrate your compounds in the well plate. And what you can do is you can measure oxygen consumption right away in cells. We know that about now 80% of all oxygen consumption in cells is due to mitochondrial activity. So if you have oxygen consumption, we can reasonably assume that this is because of mitochondrial energy consumption. Now, of course, you first got to start with, so this is when we start out, this is about the oxygen consumption. We've got to give an oligomycin, which is a complex 5 inhibitor, the inhibitor of ATP synthase. And of course, if you do that, it drops right away the oxygen consumption as expected, and then you sit with a basal level of oxygen consumption, and that is through other pathways, for instance, the uncoupling proteins that are naturally there in the mitochondrial membrane. But if we give xanthomol as low as 5 micromolar, and this is levels that we do find, let's say, in liver or muscle tissues in our treatment assay, you get now within a couple minutes, you get a profound increase in the oxygen consumption, thereby supporting the hypothesis that xanthomol works as a proton uncoupler, a very mild one, because we cannot have a strong one because that would actually kill the organism. But we know from other studies that xanthomol in animal studies is not toxic. Now I have a few minutes left, and now what I want to do is talk about de novo lipogenesis. Now, de novo lipogenesis, you get fat through intake of lipids, but you can also get fatty acids by converting sugar or sugar derivatives into lipids, and that is shown here. And if you do this in the presence of deuterium oxide, D2O, you get labeling, covalent labeling of fatty acids. Now that's very good because we can measure that quite easily with mass spectrometry. This is not anymore the metabolomics, we call this the lipidomics. And our LCMS, in 15 minutes, we can measure all lipids in no time. And if you look at a spectrum, so this is the high mass accuracy, high resolution. You see these are isotopes. See that these are far away. So this is very high resolving power that we have in the mass spectrometer, and we can measure the ratio of the isotopes quite easily. And if we do this, then we get a picture like this. So this is in the pre-adipocytes. We expose the cells to D2O and then with xanthohumol. And of course, if we don't use D2O, there is no change in the isotopes, isotopomers, right? Because there should be, because there is no deuterium. However, if you do this in the presence of deuterium, you see when you also treat with xanthohumol, you get about a 25% reduction in de novo fat synthesis. This is quite significant. And we also treated it with a positive control, and the positive control is not as active. And if you treat both together, we get even a more profound effect. Now, this method we want to use now in vivo. This has not been done, what we're thinking about is, and we have already a protocol in place to do this, and it's been approved. So this will be something for the future. And this is my last slide. And I'd like to thank the people that have done this work. Cristobal Moran in my lab, he did all of the animal studies. Nicole, she did the animal studies and looked at the classical endpoints. Jay Woo Choi did all of the lipidomics analysis, and Jay Kirkwood, who got a PhD on this project, he did all of the the quilt studies with the 8,000 peaks, and he was very good at that. And I thank also the National Institute of Health for funding the study, and I thank you for your attention.
You're right on time. So one question for Dr. Frederiksen, sir.
How about dihydroxy? Not what is necessary for your activity?
Boy, I am glad that you mentioned this now, because I had another grad student who developed a synthesis for dihydroxanthohumol.
Right.
Because the problem with xanthohumol is it is electrophilic, so that's not good. And it can also cyclize to make the 8-prenylnaringenin, giving you estrogenicity. So I asked my student, can you make the dihydroxanthohumol? And she made this with catalyst and with ionic liquid. She wrote her thesis, and it was defended successfully. And of course, on the same day, we filed for provisional patent.
Because we are working with biotransformation of that such a compound for
Yeah.
the last 7 years. 45 grams type 100% converted to this compound. And the science told me that this kind of derivative is so important for the producing another compound.
I'm willing to say now, because we have submitted our provisional patent, that in the seahorse assay, the oxygen consumption assay, the effect was much more pronounced, but we cannot say it's because of a pharmacodynamic effect. There's also, of course, higher bioavailability there. Yeah. And one last comment, we also did a fecal slurry, because the gut microbiota have activity, the reductase activity, and actually we found the dihydroxanthohumol in fecal incubations of xanthohumol. So this is something that can be made in the human body.
Can I follow up a very quick one? Actually, the protonophore that you mentioned about is very useful because one of my colleagues, the Hong Kong UST, University Scientist, is focusing on the young promoting Chinese herb, which is specifically act on ATP. Okay, so I mean, your molecule would be very interested if he can pick it up.
I think many natural products that have lipophilicity,
yes,
and they have ionizable hydroxy groups, right, meaning they can dissociate and pick up a proton at the base at the acidic side of the mitochondrial membrane. They could have that effect. There is no structural requirements for this activity. It only needs to have a lipophilicity and ionizability.
Yeah, according to Chinese medicine theory, you have yang and yin, yeah? And then he named this and published in the Trend TIP, and he said this is Chinese yang acting herb on ATP. Mm-hmm. So that model is very interesting.
Okay, another question? Okay, I will just, if you just hold on to your question, I have a suggestion. Right after Dr. Asakawa presents the certificate of appreciation to Dr. Stevens, I would ask our guests to stay on the podium, and if there are any questions from the audience to the panel, we'll have a few minutes to do that, and so it will be an open discussion, maybe for 5 minutes or so. So at this point of time, yes, we can spend maybe a few minutes for the audience to ask questions, address the panel with whatever on their mind. So please.
Hello, good morning. I want to ask to Dr. Frederik that can you explain the seahorse extracellular flux analyzer experiment, and what kind of samples from the rat you have used for that experiment?
Now, in the old days, you've got to isolate mitochondria and do with the Clark electrode measure oxygen consumption. Now, this method was hated by people who work with it because to isolate mitochondria is already a pain in the neck, I should say. Okay, I say the other one. Now this seahorse flux analyzer, what it is just a computerized system, and it works with cells. And of course, it works on the assumption that most of the oxygen consumption is actually taking place in the mitochondria. Now that is a compromise, but let's say 80–90% of oxygen consumption is taking place in the seahorse, in the mitochondria. So what you do is you plate your cells in a 96 volt, and then you fill your syringes with your test compounds, and then actually with a computer you can titrate your compounds to the cells. And what it does is electrodes, they come down into the cells and they measure oxygen consumption through a fluorimetric analysis. Now I didn't mention about that, but another thing that you can measure is called extracellular acidification. It actually measures the excretion of lactic acid into the media. There you can measure actually glycolytic activity. So they called in the old days, actually it was the Warburg effect in cancer cells. So there's two endpoints that you can take. You can measure oxygen consumption and lactate production in whole cells, computerized automatically. So students set it up, and they push the button, and every so minutes you can see the date, and you get a curve like this that I've shown. I can tell you it would cost about $130,000. Cheaper than a master tom.
But which kind of the cells you have used? You have isolated the
Oh,
cells
these
from
are,
the plated rat
I
or
forgot to
cell
mention,
lines?
these are, of course, you've got to take cells that have most activity. So where is energy consumption taking place? Is in the muscles, right? But there is where all the energy expenditure is. So we use mouse skeletal C2, C12 cells. And I'm sorry that some misprint happened in my abstract because it was the C2CO2, which is an impossible chemical compound. is not, it just means C2, C12. That's the type of the cells, and that is readily available. You can buy the cells from any source, any cellular source. Very useful compound, but please remember, it will only confirm data from another method, right? Because by itself, it doesn't tell you. So that's the thing to keep in mind. But I think the combination of metabolomics with a orthogonal method for this is actually quite useful.
Confirmatory.
Confirmatory. If you publish a paper with only metabolomics data, most editors won't say they don't want it. You've got to confirm it with a functional test. Now this qualifies as a